Alrighty, welcome to virtual class while we are all snowed into our houses or apartments or wherever you are. Hopefully you are safe and warm. All right, so to keep us moving, because we need to keep going through this unit, we are gonna do some notes on here again. I'll post the slides like I always do. So your assignment is going to be to write down the notes in your notebook or wherever you put them. I realize you don't have the, diagrammed ones like where we can label the different structures we're talking about today. I will get those to you when we get back into physical in person in class, and you can just transfer the information over. For now, just do your best on a piece of paper. So your assignment is to write down the notes, watch this video, and then you'll upload a picture of your completed notes for your grade essentially for the day. I'm not looking for Picasso structures or anything like that. I'm just looking to show you did it and you sort of paid attention? Question mark. So we're going to go through a couple smaller slide sets today. So we're talking about the structure of the sarcomere and the structure of the myofilament. So there'll be two sets of slides on your Google Classroom. We're going to start with the sarcomere. All right. So you've seen this image before in some of our previous notes. If you don't remember anything from the previous notes, pause this, go look at them. Remember, we talked about the different components of the muscle going from, where the tendon connects it to the bone, to the actual muscle structure itself. Remember how it's layers on layers and layers, and there's connective tissue around each layer. Remember, we're going from the largest to the smallest. Okay, in those notes we went over, you saw a picture kind of like this, where we talked about like actin and myosin. And I wanted you to know those were myofilaments and proteins essentially. Now we're going to get into the different components of this particular structure. And then we're going to talk about the other myofilaments in general and how they work together. Because next week we're going to talk about what's called the sliding filament model, aka the cross bridge cycling, whatever you want to call it, same thing. And that thing is not going to make any sense to you if you have not been keeping up with the material so far, especially with how these things interact with each other. It's just not going to make any sense. Okay, remember I told you the first time even I learned this, I was bamboozled. You got to pay attention to details and stuff on this. Don't just try and wing it the night before a quiz or a test. It's not going to go well for you. Okay, please don't do that. All right, so background. Remember, muscles contract, they relax. If they're contracting, they're getting shorter, right? So what's happening when this contraction is occurring is these tiny little sarcomeres, these little structures right in here, they are actually contracting themselves. So the whole process of you picking up something, your muscles contracting in your arm when you do this, the whole process is because those sarcomeres are working together and they're all contracting at the same time. So if you have things that are not like, we talked about the neuromuscular junction, controlling those impulses at action potential, making sure we send the signal down through the T-tubule so it gets all the different layers of the muscle are innervated at the same time. If that doesn't happen, that's why we're not going to have contraction happening at the same time, and that's going to cause the movement to either be very weak, non-existent, kind of jerky and floppy. Like that's not effective, right? If you're trying to kick a soccer ball into the goal, you want to know your muscles are all going to contract properly at the same time. But when I say that, I mean that sarcomeres, the structure inside the muscles, are all going to work together to contract, and that the ball is going to go into the goal because your muscles in your leg did what they were supposed to do. All right, so sarcomeres are made of myofilaments. We kind of talked about that a little bit in one of the previous notes. I don't remember which one. All right. So myosin, we've talked a little bit about myosin. So this is the thick myofilament. It has what we call these little heads, kind of these little nubby things. They bind to actin. So myosin binds to actin, okay? Myosin is going to play a big role in the whole process of muscle contraction. So you pay attention, okay? Because what's happening here is it's going to bind to actin, as we'll see a little bit today. It's going to bind to actin, and when this happens, it's pulling on actin, and that's going to cause the sarcomere to shorten, okay? So then we have actin itself. You notice it's pointing to this little kind of rope up here. So this is where it kind of looks like two strands of pearls, if that helps you remember kind of which one is which. So myosin's going to have that kind of little like nub sticking out, that little head as we call it. And then actin is going to be these little pearl strands, if that helps you remember it. Okay. So we're going to talk about the different parts here. Actin is attached to something called the Z line, which we'll get in that in a minute, but it's this little ziggy-zaggy over here. If you notice, it kind of looks like a Z. So if you'll notice, actin is attached to it. This is important, okay? So part of what is causing this contraction is this whole sarcomere shortening, right? And you're probably going, okay, then everything inside of it also shortens as well. Not necessarily. So these little grabby guys, these little myosin heads are going to grab onto the actin. Neither actin nor myosin are actually getting shorter in length. They're just grabbing onto each other and pulling. Okay, it's like a resistance. Okay, think about like tug of war with, the rope. Both teams are pulling on the rope, but the rope is not getting shorter, right? Okay, so here's that Z line. Basically, the Z line is a fancy name for the boundary between sarcomeres, because sarcomeres are going to be lined up one after the after another, and they are kind of touching essentially, kind of like how train cars all line up. So if you have this Z line here, you can see it on this little cartoon guy here. Here's your Z lines. Here's your Z line. So everything in between here is one sarcomere. Okay. It's when we're looking at like muscle fibers under a microscope, if we're trying to figure out if something is correct or incorrect or whatever for the structure, we're going to look for certain features on these different sarcomeres to identify what's what and where it is and what's going on with the function. So when a contraction is happening, so if you'll notice on this picture, we've got a Z line here, we've got a Z line over here. So here's like another sarcomere would be over here. When this happens, this little head of myosin grabs the actin, they're pulling against each other, okay? So the sarcomere itself will get shorter, and when that happens, these Z lines actually get closer to each other. Okay, so you would see the Z lines come closer together during contraction, and they get farther apart during relaxation. All right, M line, aka the middle line. Guess where it is? It's in the middle. All right. So this is going to hold together the different myosin myofilaments. In this picture here, it's these little red lines here. And guess what? The middle. Oh my goodness, it's amazing. It's in the middle. All right, so here's our Z lines. Here's our Z lines. So it's right in here on this kind of dark band on this cartoon right here. Okay. When the sarcomere is contracting, we're going to have the actin and actin. They're going to meet there in the middle at the end line. Okay, just like kind of tug of war, your middle point, wherever that rope or whatever the point is, it's going to meet somewhere when the contraction happens. Okay, so actin are going to meet together. All right, and then we have something called an I band. So an I band is only going to have the thin filament actin involved in it. So if you'll notice, actin crosses over some of the myosin, but it also has spaces here where there's no myosin underneath it in this relaxed sarcomere. Okay, because remember, it would get closer together. So the I band is going to be this section right here, or over here on this side, respectively, whichever you're looking at. And it is only actin. Okay. If it helps you to remember the I band, light has I in it, thin has I in it, actin has I in it, light, thin, actin, if that helps you remember I band, whatever mnemonic helps you, because I know some of these are going to be a little confusing at first. So if there is any myosin involved in any area of this, it is not an I band. Okay, it is only actin. Make sure you know that. If you remember us talking about the different kinds of muscle tissue, remember we talked about skeletal, cardiac, and smooth muscle. Remember, smooth muscle is distinct because it doesn't have striations, it doesn't have the stripes, but cardiac and skeletal do. So those striations, those stripes, when you look at them like under a microscope, what you're seeing is dark and light bands, essentially, which is where these come in. Okay, so the light bands are the I bands. So if you look under a microscope and you're trying to see what kind of muscle tissue you have, one of the things you immediately look for is, does it have striations? That's a dead giveaway of what possible your ones you're looking at. We haven't talked about it yet, but cardiac muscle would have intercalated discs. That would be the other dead giveaway. Okay. Then we have the H zone, which is where we only have myosin. So you notice we have areas where we have both, areas where we have only one, which was the I band. Now we have this H zone, which is right over the middle almost. It's not the M line, okay? But it's where there's only the thick filament, aka myosin. So do you see how there's a difference? Okay. So same thing with the other one for the I band. If there was any myosin present, it was no longer an I band. Same thing with this one. If there's any actin present, it's no longer the H zone. Okay, they have their own little respective areas. And again, this area is going to get smaller and smaller if contraction is happening. Okay, in this whole broad area here, this is called the dark band or the A band. Again, dark has A in it, like light has I in it. So again, it's going to be the other part of that striation. So the I band, the light band was for the lighter stripies, the striations, and then this is for the dark striations, the other counterpoint to it. So this is where it's going to be what we define as the entire length of a myosin filament. And if you'll notice, it's got actin and myosin in it because it's covering this whole area. So this actually, the other two, the H zone and the I band, they change size based on if contraction's happening, right? This one does not change size regardless, okay? Because myosin itself doesn't change in length. So remember in your notes, write down that this one does not change in length for contraction, but the other two do. The I band and the H zone change in length for contraction. The A band does not. Make sure you know that. All right. So then we're going to pivot to the myofilaments. One here. All right, so myofilaments. So there's a couple of other new terms you're going to see in this one. Oops, that was squeaky. So let's go ahead and get into it. All right, so again, this is a review. Remember, the sarcomeres are contracting, the muscle's getting shorter. We talked about sarcomeres being made up of myofilaments, specifically actin and myosin. Remember, we are pulling when contraction is happening. I can speak, I really can't. Myosin is pulling on actin to get it towards the M line, that red middle line there. Okay. So again, we're just lumping this all into one, but remember, we just talked about it. So if you're still not sure of what's what on a sarcomere, you've got to go back, look at the pictures. Again, draw out this little sarcomere in your notes and label each section. That's the best way to learn which is which. It's one thing to just kind of look at a slide and be like, yeah, that makes sense. I know that. Actually draw it out and label it. All right, so we know this. We've talked about this a few times before. Remember, actin and myosin, they're myofilaments. Myofilaments make up the sarcomeres. Remember, you drew that out in your project. Okay. All right, so myosin. So now we're going to look more specifically at some of these. And again, I don't have the printouts, lovely diagram for you to do this with me where it says, color different things. So when we get back into in-person, probably Monday, maybe Friday, I don't know. I will have these for you and you can color them in and put them in your notes, okay? So myosin, we know it's a thick filament. It has a whole bunch of those little myosin heads, okay? And they really want to bind to actin. That's what their favorite thing in the world is. And to do this, we need ATP. So ATP, if you don't remember, we talked about it a little bit, but it's essentially cellular energy. It's adenosine triphosphate. Your body generates this ATP through chemical reactions where you consume food for your energy, right? You consume proteins and sugars and all that good stuff. And it turns it into ATP for your body. That's why you can do anything, is because you have ATP powering your cells, powering these different reactions, like the myosin grabbing onto actin. Okay, if there is no ATP, there's no muscle contraction happening. Okay, so muscles, especially your skeletal muscles, remember they're voluntary. They can't just go and go and go and go and go and go and go like your heart muscle can. So that's why they get fatigued. They run out of ATP. if you've ever played a sport or done anything that requires a lot of energy, you know this. Eventually, you will run out of energy. Your muscles will get tired. They will get fatigued. That means they have run out of ATP, which means there is no more myosin binding to actin in that particular moment. Now, you can get rid of fatigue, you can get some more energy, but your body has to remake it. So that's why if you are in a sport, especially if it's like a long tournament, regionals, something, an all-day event, right? And you're off and on, off and on, off and on. Like I was a swimmer, right? So I might have an event in the morning. I obviously ate breakfast. I was good. So I had fuel going in my body that was being turned into ATP. So my muscle cells could use that so I could, swim. But swimming is a very intense sport, especially if you're a sprinter. I wasn't. I was a distance swimmer. So you have to restock on that ATP, that energy source. Well, it doesn't just come from the sky. So after my first event, which is usually like the 200, I would then go over to my little snack bag and go have a granola bar or protein bar or something like that because I needed to re-ingest food so that my body could turn that food into the chemical energy signature that is ATP. So that way I could then do my next event, whatever it was, right? So if you ever in a sport like that, that's why your coaches are pushing you. Well, not, hopefully not pushing, but again, you know what I mean? They're like, hey, you need to hydrate because you sweat out a lot of water and electrolytes. That's why Gatorade is so popular. And it also has sugar in it. It's another reason. But they're also like, hey, go eat a granola bar, you know, get some fuel in your body, especially if you're in a very high taxing sport where you use very large amounts of energy very quickly, like sprinting and track or swimming or whatever it is. So you're going to use large amounts of energy very quickly. You've got to refuel that tank before you can do your next event. Or if you've never refilled your tank, you probably saw this. You didn't do so good on the next event because your muscles were fatigued. They ran out of ATP. They're like, we're done. We're not doing this. We're done. All right. I got on a tangent. Or was I? I don't know. Okay, so there are in your myofilaments, there's about 300 myosin molecules all bundled together. That's a lot. But I mean, think about how much energy and structure is needed to actually like play a sport, right? This makes sense. So the tails of the myosin, these little tails guys, right? They are going to form the central part of the thick filament. And then the heads kind of face outwards. You can see from the structure here. So if you look at this picture, that when they're relaxed, which means this myosin head is not bound to actin. It's just they're just chill. They're here. And then we've got the actin up here, the little pearl strands, right? Now when we're contracting, we're using ATP. These myosin heads, they are like, oh yeah, I get to bind to actin. It's great. And so they reach up and they grab and they pull. Okay. So be able to recognize, like if I put one of these pictures on a quiz and said, is one, what's involved here? Is it myosin? Is it actin? Is it who is it, right? And be able to tell me which is relaxed, which is contracted, which is energy using and which is not. Okay. All right, so that's myosin. Then we have actin. Remember, it's our thin filaments, the pearl strands, if you will. So we've got basically actin is two different, what we say is it's going to be two different myofilaments twisted together. That's where we get this pearl thing. And the whole main reason here is we've got these binding sites for the myosin specifically on it. Okay. And again, we notice, again, here's our myosin down here for relaxed. Then it'll grab on to the actin, which is these little purple dots, the little pearls. Okay. And then when that happens, we are contracting. We are using energy. Okay. It does have regulatory proteins that we're going to get into to kind of control the chaos. Because you don't want this just randomly happening and, wasting energy and firing off contractions, because if that happens, there's a chance maybe you're not in control of your muscles, which can happen in certain diseases and situations. That's why we understand these different structures at the cellular molecular level of the muscles, because that tells us a lot about what is happening and why in certain diseases where people maybe can't move or they can't control their muscle spasms or things like that. So one of these regulatory proteins, so to speak, is tropomyosin. Tropomyosin. So it's kind of like a rod, if you will. On this picture here, you will see it's kind of like this blue line right in there. Okay, so actin needs a whole lot of support to do its job. I mean, it's there to attract the myosin, but it needs some supports. Okay, so otherwise it's like a wet noodle. Like, have you ever seen it, wet spaghetti noodle? It's you can't make that thing stand up on its own. It's no. So this tropomyosin comes in and helps stiffen and stabilize that actin so it can be there in the right spot at the right time for the myosin to grab hold of to pull. Um, and this is also going to kind of act as a mediary in between myosin and actin when there is no contraction happening. Because remember, myosin and actin really, 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 really want to bind together. That's all they want. But you can't have your muscles contracting all the time. Okay? That uses a whole lot of energy, and if you've ever had like a muscle cramp, that's not comfortable. So this guy, tropomyosin, he's going to come in and block those myosin sites so they cannot bind if if the muscle is relaxed. So there's no contraction. So tropomyosin comes in and says, "No, you can't come in here right now." All right. So when you're looking at a picture, so tropomyosin is going to be kind of this spirally thing going all the way around. Okay, so make sure you know that on a picture. So if you'll notice, see how it's kind of on the relaxing here, it's kind of blocking the pearl strand. Okay, so it's saying the myosin heads can't grab on. We're blocking it. Now there's contraction happening. We're using ATP. Myosin heads can grab the little actin pearls because look, tropomyosin here in green has moved out of the way. So, I mean, obviously it's still there. It doesn't just cease to exist, but it's moved out of the way. So now the myosin head can grab onto the actin. Okay. So you kind of see how it's kind of swirling, spiraling, whatever I'm trying to say, around. Why is it people always want to talk to me when I'm doing something? Okay. So hopefully you're following me. Make sure I'm on the right page of my notes because I'm in tangent land today. All right, so that's tropomyosin. Just because we sometimes have to be confusing, the next one is troponin. Now, how they sound very similar, just like I've told you 100 times before in this class, sometimes words look and sound very similar. Slow down and read them carefully. Okay? Don't just look at the first part of the word and think you know what it is, because you could be wrong. Okay? So this particular item in this whole process, so it is a globular protein, if you want to be real specific, and it has 3 different subunits, so 3 different components to it. So one subunit, its whole job is to bind to actin. Another one is to bind to tropomyosin. The other one, it binds to calcium. So wait, calcium, where have we heard that before? Where is that coming into play? Calcium equals contraction. Hopefully you remember that. I wrote that on the board. I pointed to it. I said this is important. Write this down. Hopefully you did that. Okay. So on our lovely picture here, here's a little orange guy. This is troponin. All right. So it's another regulatory protein. It's helping control this whole process because remember, myosin and actin just love each other so much. That's all they want to do is just bond and pull. Okay, that's all they want to do. But we need certain mediators involved here. And one of them is tropomyosin and one of them is troponin. So the way I keep these straight, because you notice the names are very similar. So you can just remember from these little cartoon structures which is which. Good for you. So tropomyosin is a longer word and it's the longer structure. It's that green, long, stringy structure, right? Troponin is a shorter word and it is this smaller structure. Smaller, shorter, longer word, whatever works for you. Okay. Okay, so we have calcium ion. Calcium's going to trigger contraction. When that calcium ion binds to troponin, our orange guy down here, Okay, so calcium is trying to show this. I think it's calcium down here. All right, it's going to pull the tropomyosin, okay, the long stringy guy, off of the binding sites for the myosin. Remember, tropomyosin is blocking those binding sites from allowing the actin and myosin to be besties and cause a contraction, right? So remember how I said calcium causes a contraction. Calcium causes contraction. Here is why. Okay. So the calcium ion, remember it's Ca2 plus, it comes in here. It's going to bind to the troponin here in orange. It's going to then pull, because you see how they're kind of connected. So we bind to the calcium to the troponin. It's going to pull the tropomyosin off of these myosin sites on the actin. So myosin can actually get to the actin and cause that contraction. Okay, let's go through that again, because I know it's a whole lot of words. You're going, what? Okay, so remember, actin is the purple pearls. Myosin down here with the blue little heads, they want to be with the actin and the purple pearls. Tropomyosin is this thing in green. It says no. During relaxation, no, you can't come here. Okay. So this is relaxation. We're blocking off those binding sites. These two cannot be besties in pool. No, we're done. Okay. Well, here comes some calcium. That calcium ion. It's going to then come and bind to this orange guy, that troponin. Troponin is connected to the tropomyosin, the green band that's blocking the myosin from getting to the actin. And it's going to pull it back. Okay. You pull it back. Suddenly, when we're contracting, now the myosin and the actin can be besties and they can bind. They can cause that contraction. They can cause the sarcomere to get shorter. Okay. I think that was the end of it. B12. Okay. So make sure you know those steps in order of who is doing what and why. Okay. So again, because this is a video, you can go back and rewind it. I don't have to re-explain it the 100th time, hopefully. But obviously we'll go through this again in class. I will get you the printouts when we go back in class. You can color them in if that helps you. Understand what is happening in what order and why, because when we get into the cross-bridge cycling slash sliding filament model next week, which is basically a cycle of all of this happening over and over and over again, explaining what's happening with the muscle itself, you've got to understand the difference between troponin, tropomyosin, actin, myosin, all of those. Okay, you have to. So make sure you write out your notes. When you have a completed set of notes from both of these slides, by the way, from this whole video, not just from one, upload a picture of it to the Dropbox on Google Classroom. Make sure I can actually see it. Make sure I can see all of it and read it. And that's your grade, easy 100. Easy 100. Okay. And I will be in touch on Google Classroom. If you have any more questions, you know where to find me. All right, guys. Hope you stay safe and warm. And have a good rest of your day / snow day / week. I don't know. I will see you soon.